The cell has another way of moving particles across the membrane that we haven't talked about yet. It's called bulk transport. B-U-L-K. And it's when the membrane engulfs an object. Takes an object, let's pretend this is something that the cell is trying to swallow. And this object might just be too big for the cell. So the cell can kind of wrap its membrane around the object and pull it in. Engulf it and pull it in and swallow it up. That's called bulk transport. You can see here in this picture, here's some large particle, and the membrane, this is the cell membrane, this orange thing. The membrane kind of moves and wraps itself around this particle and pulls it in. And then it'll pinch off the membrane around the particle. Now the membrane is containing the object. So in this case, it looks like a bacterium. And we call that a vacuole if it's big, and we call it a vesicle if it's small.
What's the connection between the large particle and the cell? Like, how does it know you need this particle?
That's a good question. Sometimes the particles have little proteins on their surfaces, and there are receptor proteins on the membrane that have the right shape, and they'll stick to it like a key fits a lock. So if it's just the right protein on the surface of the object, it'll stick to the protein on the surface of the membrane, and then the membrane reacts by surrounding it. That happens sometimes. There's probably thousands of ways the cell can figure that out, but it probably usually has to do with protein surface receptors. And there's probably ways we don't know about. Maybe it feels pressure, water pressure or something pushing up against it, and it goes around it, I don't know. There might be other ways. Maybe something electrical, or ions coming off of a bacterium or something. There's probably a lot of different ways to sense things. It's a great question. And scientists are learning how it works every day. Would you like to see video footage? This is called phagocytosis, sometimes known as phagocytosis. When you surround a large particle and pull it in, it's phagocytosis.
Phagocytosis, which means cell eating, is a form of endocytosis. A cell such as a phagocyte engulfs surrounding material, forming vesicles called phagosomes, derived from the plasma membrane. Phagosomes merge with vesicles called primary lysosomes that contain digestive enzymes, forming secondary lysosomes. Digestion takes place within the secondary lysosomes.
This is how your white blood cells eat bacteria and kill them. More video footage.
In phagocytosis, phagocytes are attracted to the area of invasion by chemical products of the microorganism, phospholipids released by injured mammalian cells, or by components of the complement
Chandler, this shows what I was talking about. Check it out.
System. The phagocyte moves into the area of invasion and then attaches to the microorganism. The attachment is mediated by a variety of surface receptors, including antibody, lipopolysaccharide, and complement receptors. For example, the C3b component of complement coats bacteria or other particles and then binds to C3b receptors on the phagocyte. This process of coating to enhance phagocytosis is called opsonization. The microorganism is then engulfed by the phagocyte into a vacuole known as a phagosome. Vesicles in the cytoplasm called lysosomes fuse with the phagosome, releasing digestive enzymes such as lysozyme and proteases into the phagosome. The structure resulting from this fusion is called a phagolysosome. Inside the phagolysosome, microorganisms are killed and digested. Finally, the digested contents of the phagolysosome are eliminated from the phagocyte by exocytosis.
Cool, pooped it out. Whoop. 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 Fun. It's fun to toggle. Isn't that cool? Okay, there's phagocytosis, taking in large particles, and there's penocytosis, taking in small particles. Basically works the same way. It surrounds the particle and pulls it in. Also called penocytosis, sometimes called pinocytosis. I like to say penocytosis.
Pinocytosis, which means cell drinking, is a form of endocytosis. A cell brings in droplets of the environment by pinching them off with the plasma membrane. The pinocytic vesicles break away from the membrane and move to the cell's interior.
What you think?
Pretty cool.
Wait, I got another one.
Cells also use energy to transfer materials in bulk, but this time by forming membranous sacs that hold their contents under wraps. In a process called endocytosis, part of the surface membrane encloses the material, forming a sac which brings the contents into the cell's interior. In the opposite process, called exocytosis, the sac moves through the cytoplasm to the membrane, fuses with it, then releases the contents.
Cool. That had trippy music. This is probably the coolest term this chapter. Receptor-mediated endocytosis. Can you say receptor-mediated endocytosis?
Receptor-mediated endocytosis.
You say that around someone, they'll think you're smart. Or they might beat you up. But either way, you're talking about when particles from the environment stick to the cell, and here you can see some protein receptors on the surface of the cell, and they stick to, looks like the star-shaped things are what the cell wants. So they'll stick to these receptors, and when you get enough of them stuck to the receptors, it forms what's called a coated pit. So when it sticks on this side, there are proteins called clathrin that come up from the underside and stick on the underside. And so the more of these stars that are received, the more of these clathrin proteins attach. And it's almost like the clathrin proteins are weighting down the area. And it kind of pulls the area down and makes it go into a bubble. And we call the bubble now a coated vesicle. Why is it called a coated vesicle? Because it's surrounded with these clathrin proteins. It's coated with them. And it contains several of the items that attach to the receptor. That's kind of a common way cells can bring in things they need. Video footage of that. Receptor-mediated endocytosis. It's my favorite kind of endocytosis. What? Start over.
In this example of receptor-mediated endocytosis, a cell takes in a particle of protein, or LDL, from the outside. To do this, the cell uses receptors that specifically recognize and bind to the LDL particle. The receptors are clustered together. An LDL particle contains 1,000 or more cholesterol molecules at its core. A monolayer of phospholipids surrounds the cholesterol and is embedded with proteins called ApoB. These ApoB proteins are specifically recognized by receptors in the cell's membrane. The receptors in the coated pit bind to the ApoB proteins on the LDL particle. The pit is reinforced by a lattice-like network of proteins called clathrin. Additional clathrin molecules then add to the lattice, which eventually pinches off a part of the membrane. This internalized vesicle is referred to as a coat.
What you think? All right. Now this is called exocytosis. It's pretty simple. It's just the reverse. A vesicle releases stuff to the outside.
This is a cell that secretes large molecules that are unable to pass through the lipid bilayer of the plasma membrane. They leave the cell instead by a process known as exocytosis. After production in a system of membranes called the endoplasmic reticulum, the molecules are packaged in a small sac of membrane called a vesicle. The vesicle moves to a stack of membranes called the Golgi apparatus.
Watch the video.
Their membranes merge, and the vesicle releases its contents for modification. As the molecules leave the Golgi apparatus, they are again packaged in a vesicle, which moves to the plasma membrane. As membranes merge again, the contents leave the cell without actually crossing the plasma membrane.
Ah, see? Exocytosis.
This process can also occur in reverse, allowing large molecules to enter a cell. This is termed endocytosis.
Ah, that shows it really well. Another video.
The endomembrane system consists of the endoplasmic reticulum and Golgi apparatuses. Vesicles pass from one part of the endomembrane system to another, and secretory vesicles from the Golgi apparatus merge with the plasma membrane,
Watch the vesicle on the
secreting the
bottom.
contents by exocytosis.
Watch it. Exocytosis. Huh? Are y'all getting this?
Yeah.
Incredible.
Protists, such as paramecium, sweep food particles into their gullet, trapping the particles in a food vacuole. Digestion takes place within the food vacuole, and the products of digestion pass through the membrane of the food vacuole. Finally, the food vacuole merges with the plasma membrane, expelling waste products into the environment.
Ew, it went to the bathroom. Understand bulk transport?
Yes, sir.
By the way, bulk transport does require ATP. These things just don't pinch in and move on their own. Just moving a vesicle through the cell requires a bunch of ATP to get that vesicle to move. Remember those little motor molecules that are walking along in the cell?
Yeah.
That requires ATP. So anytime a vesicle moves, it requires ATP. Okay. You can stop filming.